Let's jump straight into this high-yield, hyperdense histological explainer of the accessory organs of digestion. We are going to map microscopic structure directly to macroscopic function and, of course, to board-relevant pathology. No fluff, just the dense academic value you need.
We're taking a top-down anatomical gradient today. We'll hit the histology of the salivary glands, the exocrine pancreas and ducts, hepatic architecture and lobules, the sinusoids and biliary tract, and then tie it all together with an accessory gland network summary. We are tracking this chemical manufacturing and secretion network sequentially. Starting off with Section 1, the histology of the salivary glands, we begin right in the oral mucosa with the major salivary glands, which operate as your very first biochemical processing stations. You absolutely need to drill these distinct structural and secretory differences among the three major pairs of glands into your memory. The parotid gland, it features exclusively serous acini, heavily secreting alpha amylase to kickstart carbohydrate hydrolysis. Then we have the submandibular gland, which actually produces 2/3 of all your saliva via mixed but predominantly serous acini, and it adds a lysozyme to hydrolyze bacterial walls. Finally, the sublingual gland is primarily made up of short tubules of mucus cells, packing its clear cytoplasm with hydrophilic mucins for essential mucosal lubrication. Let's visually anchor these distinct cellular features. On the left, look at those densely packed, strongly basophilic serous acini of the parotid gland. Notice those central round nuclei and the really dark staining. That indicates abundant, rough endoplasmic reticulum. Now contrast that with the submandibular gland on the right. Here, you can clearly identify pale, staining mucus tubules, and they are capped distally by distinct, crescent-shaped clusters of serous cells. We classically call these serous demilunes. Now, understanding the ductal modification system is a crucial point. Secretory acini empty into short intercalated ducts lined by cuboidal epithelium, which then feed directly into the striated ducts. These columnar striated duct cells have heavy basolateral membrane infoldings that are absolutely packed with mitochondria. And that massive mitochondrial concentration is non-negotiable. It actively powers the rapid transcytosis needed to reabsorb sodium and chloride while adding bicarbonates. This fundamentally modifies the primary saliva into a hypotonic, buffered secretion. Notably, mutations in chloride channel transport here, and similarly in the pancreas, drive the thick, obstructing secretions that are highly characteristic of cystic fibrosis. Let's integrate some high-yield pathology right into this histological framework. When this secretory architecture is compromised, say by a mump's viral infection or as a pharmacological side effect of antihistamines, you get xerostomia, or dry mouth. That completely disrupts your mucosal protection and early carbohydrate hydrolysis. On the flip side, an autonomic overdrive from nausea or even a rabies viral infection triggers sialorrhea, which is an excessive, uncontrolled production of this fluid. Moving to Section 2, the exocrine pancreas and ducts. As we head inferiorly into the retroperitoneal space, we transition to the absolute major producer of our digestive enzymes. You must instantly recognize centroacinar cells. This is a pathognomonic, ultra-high-yield histological identifier for the pancreas that completely differentiates it from the parotid gland. These pale staining cells represent the very beginning of the intercalated ducts, and they insert directly into the lumen of the acinus itself. Under the stimulation of secretin, they secrete a massive volume of bicarbonate-rich fluid to really alkalinize that ductal environment. Just look at the extreme polarization of these pancreatic acinar cells. See the intense basophilic staining at the basal pole? That's due to the massive concentration of rough endoplasmic reticulum required for heavy protein synthesis. But apically, the cytoplasm is strongly acidophilic. It's packed with membrane-bound zymogen granules storing inactive digestive enzymes right at the luminal border. Because the pancreas produces such incredibly destructive proteases, lipases, and nucleases, it relies on three strict biochemical safeguards to prevent catastrophic autodigestion. First, proteases are exclusively secreted as inactive zymogens, like trypsinogen. Second, they're co-packaged inside those apical granules alongside highly specific trypsin inhibitors. And third, the centroacinar and intercalated duct cells maintain a highly alkaline intraductal pH via continuous bicarbonate secretion. This keeps the enzymes dormant until they safely reach the duodenum. But when these structural safeguards fail, pathology is immediate. The premature autoactivation of zymogens, often triggered by gallstones or alcoholism, results in acute pancreatitis, which rapidly digests the sparse connective stroma of the gland. Also, keep in mind that pancreatic cancer typically arises as a carcinoma of the ductal epithelial cells, most frequently localizing in the head of the pancreas, aggressively metastasizing through that surrounding connective tissue.
Let's tackle Section 3, hepatic architecture and lobules. We're transitioning to the largest internal organ, the liver, where we have to master the complex microarchitecture of the hepatocyte plates.
To truly master the liver, you must conceptualize 3 distinct models. The classic hepatic lobule model emphasizes endocrine function, with blood flowing radially inward to a central venule. This allows hepatocytes to extract toxins and nutrients while simultaneously secreting newly synthesized plasma proteins, like albumin, right into the systemic circulation. Conversely, the portal lobule model emphasizes exocrine function, tracing bile drainage outward to the portal triad. Finally, the hepatic acinus of Rappaport divides the functional parenchyma into 3 metabolic zones based strictly on oxygen gradients. The portal triad, located out at the periphery of the lobule, is a non-negotiable structural cornerstone. It contains 3 distinct vessels. You've got a bile ductule, an arterial branch of the hepatic artery supplying 25% of the highly oxygenated blood volume, and a venule branch of the portal vein supplying the remaining 75%, which is completely loaded with oxygen-poor but incredibly nutrient-dense splanchnic blood. These 2 blood supplies perfectly mix right as they enter the lobule. So to understand how these polyhedral hepatocytes actually manage to process massive amounts of toxins, synthesize vital plasma proteins, and store extensive glycogen, we have to ask a crucial question. Where is the exact microscopic boundary where this mixed blood actually meets the cell? Which brings us directly to Section 4, sinusoids and the biliary tract. We're diving into the hepatic sinusoids and the highly specialized cellular network residing within their delicate architecture. The hepatic sinusoids are lined by a discontinuous, fenestrated endothelium, and residing directly within this sinusoidal lumen are your Kupffer cells. These specialized monocyte-derived resident macrophages relentlessly patrol the blood, phagocytosing effete erythrocytes, freeing up heme and iron for local storage, and clearing out portal-borne debris. Because of those endothelial fenestrations we just mentioned, blood plasma freely leaks out to bathe the hepatocyte microvilli in a microscopic gap called the perisinusoidal space, or the space of Disse. This is the precise location of metabolic exchange. Residing quietly within this space are the hepatic stellate cells, or Edo cells. Their main job is storing vitamin A and small lipid droplets and maintaining the sparse extracellular matrix. Pay close attention, here is your high-yield pathology. In chronic liver disease, those vitamin A-storing Ito cells undergo a pathological transformation into myofibroblasts. They start depositing massive amounts of collagen directly into the space of Disse. This completely obliterates the metabolic exchange interface, creating the deadly fibrotic architecture of hepatic cirrhosis. Also notice how the pericentral hepatocytes, in zone 3 of the liver acinus, receive the absolute lowest concentration of oxygen and nutrients. This makes them highly vulnerable to ischemic necrosis, which we often call shock liver, as well as toxic metabolite damage from things like acetaminophen. Now, while the basolateral domains of the hepatocyte interface with blood, their apical surfaces form tiny grooves called bile canaliculi, which are sealed tight by occluding junctions. Notice the strict countercurrent flow here. While blood rushes inward toward the central vein, exocrine bile is actively secreted into these canaliculi, flowing outward, draining into the canals of Herring, and ultimately reaching the cuboidal cholangiocytes of the portal triad bile ductules. Finally, Section 5, our accessory gland network summary. Let's synthesize this entire histological gradient into a single, unified mechanism of digestion. From the top down, the accessory organs of digestion operate as an uncompromisingly coordinated chemical manufacturing network. The salivary acini initiate enzymatic breakdown and buffer the oral mucosa. Then, the pancreatic centroacinar and acinar cells flood the duodenum with an alkaline, zymogen-dense fluid to execute major luminal hydrolysis. Simultaneously, the hepatic lobules synthesize the bile acids necessary to emulsify massive lipid loads. This bile is stored and concentrated in the gallbladder, which, remember, is a specialized sac completely lacking a submucosa and muscularis mucosae, lined by a highly folded simple columnar epithelium with prominent microvilli. Under the stimulation of cholecystokinin, its smooth muscle contracts. And crucially, if this concentration process crystallizes, you get cholithiasis, driving chronic inflammation into its deep mucosal invaginations, known pathognomically as Rokitansky-Aschoff sinuses. I'll leave you with this final analytical thought. The massive, macrophysiological success of human digestion relies entirely on the precise microhistology of these accessory glands. If just one microscopic zone fails, if an itocell deposits collagen or a zymogen activates a millimeter too early, how rapidly does that microscopic structural collapse compromise the entire gastrointestinal physiological cascade? Think about that as you review this high-yield material.